So I'll just start again by saying, yeah, it's a real privilege to be able to talk to you today about the use of this technology in crops. And to say that, although many of the new developments with the technology are made in animal and human cell systems, we can move those very easily into crop systems. And there's a huge amount of excitement in the crop research community about the use of this technology. So I want to start by showing you a plate of food. Now, this might not look very exciting to you, but actually it is a very exciting plate of food because this is the first CRISPR dinner. Now, this was created in 2015. As you can see, we've got tagliatelle with crispy fried vegetables. Now, these vegetables were grown in a garden of a researcher called Stefan Jansson in Sweden, and he cooked them up with his tagliatelle, a nice glass of wine. He invited a journalist, and he served this first CRISPR meal. He was able to do that in Sweden because the competent authorities in Sweden had decided that this vegetable, it had no foreign DNA, but it just had a small mutation, so it didn't need to be regulated as GM, so he was free to do this. And it got a huge amount of publicity at the time. I'm going to come back a bit later to this plate of food, but I just wanted to start. Apologies for doing this before lunch, but I just wanted to start with this. But just to now sort of set the scene again with the plants. So plant breeders have done amazing things. I mean, this is illustrated just by looking at the incredible range of brassica vegetables that we have that have all been developed from something that looked a bit like a wild cabbage here. This is all conventional breeding. And the reason that plant breeders have been able to make these amazing crops and foods that we enjoy today is because they've had access to variation. So this is mutations in the DNA, changes in the DNA that are useful to them, that they can select and incorporate into these improved crops. And of course, these mutations in the DNA are also incredibly valuable to us as researchers in taking forward our research programs and understanding which genes are really important in including in our improved crops for the future. And of course, I don't need to tell you that the need to improve crops at the moment is really quite urgent. We have to be able to tackle new crop diseases. We really need to increase yields. So, so variations are important. So what are the sources of variation? So very basically, of course, the main source of variation is simply from the same or closely related species that you can cross together. And this is what's been done over many, many years. Sometimes you can get variation in other ways. Sometimes if you cycle something through tissue culture, you introduce variation. A technique that's been very widely used, that I just want to mention briefly, is something called mutation breeding in crops. This is actually, it dates back to the 1930s, 1940s even, where seeds are treated with either chemicals or with radiation to deliberately make changes in the DNA. Now, this is a conventional plant breeding technique, but it is a way of creating variation in the DNA, and actually there are about 3,000 varieties that have been registered and commercialized that have this mutation breeding step in their pedigree. So, and I want to mention this because obviously we can make a comparison between this technology and what we can do with gene editing. Now, of course, if we've got a gene that we want to introduce that we simply can't cross in, then we have genetic modification methods. We show here the two methods that are commonly used in plants, the gene gun and the bacterial method of introducing genes, which is agrobacterium, a soil bacterium. But then we have a whole raft of what we call new breeding techniques, and that includes genome editing, of course, that's being the most exciting one of them. And this now gives us a new way of creating precise variation that could be of real value to plant breeders. So I put this slide in again just to make a comparison, because we've talked about where does GM and gene editing, where's the dividing line? And it's a little bit complicated, and we can see this in plants, because so this is just illustrating traditional breeding. So if we take an elite variety and a related variety, and we want to transfer this gene with the cross maybe. So we cross these two together, and it's almost like taking two huge stacks of playing cards and shuffling them. And you end up, yes, you've transferred what you want, but you've got a lot of other stuff that you don't want. So the breeders have a really difficult task in sorting this out and back-crossing to get back to something which is close to the elite variety but contains the extra gene. And actually they can never get that precisely. They'll have a lot of other unwanted stuff that will be tagged along as well. Now, of course, if we did that by GM, we'd just be transferring the gene that we wanted hopefully into the elite variety, or there are some challenges doing that. But one thing to remember is that that new gene would be inserted in a random location in the plant DNA. Now, we can actually do something very similar with gene editing, which is where the confusion comes, because we can also insert a new gene with gene editing, but the difference will be that that new gene will be inserted in a precise location, so we will know exactly where it is. And as has already been described, we can use gene editing to replace an existing gene with a slightly different copy. But the main way that we're using gene editing at the moment in plants is this, what we call this type 1, and this is what's been described where we're causing a double-strand break, and the cell's repair machinery is kicking in, making a repair and making a small error, so there's a small mutation. And this is the main way that we've used it so far in crops, and I want to show you a few examples. So this just shows some of the crops that we work with in my lab, and I would say that in all of these, the gene editing is working incredibly well to make these small targeted mutations. And I'm going to just pick one or two examples to show you. But one thing that I want to mention at this point, we're thinking about the challenges of this that still remain for this technology. So we're now dealing with plants. Plant cells have a cell wall, which is quite impenetrable. So we have a problem with delivery of the components for them to make the editing happen. And that's something unique in plant systems. So often we need to go through a standard genetic modification process to get all the components in, and that usually involves a tissue culture regeneration process. And you can see some of the steps of the regeneration process here. So in the cereals, wheat and barley, we start from tiny immature embryos, from immature grains, and these go through a callus induction regeneration process to get the plants back. And if there's a bottleneck anywhere, that's where it is. Because there are not so many people now that have these tissue culture skills. It was considered rather unfashionable, but now everybody needs to do this again. So these skills are really in demand again. So if any students who are thinking of tissue culture skills, very, very important. So I see, when I'm sort of looking at, if you like, bottlenecks in the system, I see the tissue culture regeneration systems and the ability to do the genetic modification steps as a real bottleneck. And because that's made worse, we've mentioned, we have now crop genomes widely available, so there's a much greater need to validate gene function. For example, we used to have to spend weeks assembling the DNA constructs to put all the components in for gene editing. Now we can do most of that with robots, so all of these barriers are being removed, but it just means that everything feeds down to this tissue culture regeneration bottleneck. So I just wanted to mention that. So there's a huge demand for gene editing in the research community. I'm just illustrating that by showing you. So one thing that we do is we offer a community resource to make targeted knockouts in crop plants. And this is funded by BBSRC. And this just shows you so we've had a couple of calls for the research community to put in their target genes to this resource. And this just shows you the numbers that have come in. So a total of 126 target genes that the research community from a lot of the different universities and research institutes would like knocked out. Unfortunately, this is over double what we can actually cope with. We're trying to do 60. So it just illustrates the demand for this technology. Now I want to show you some specific examples. And the first example I want to show you is one of the first gene edits that we did. This is one technology that I think, amazingly, it just worked beautifully the very first time we tried it. And so this is an example in a brassica crop. So in the brassica crops, the seed pods have a habit of shattering quite easily. Now, of course, if they open and they shatter too soon, the seed ends up on the ground, the farmers are not able to harvest it. So there was a Lars Ostergaard, who leads a group at the John Innes Centre, is particularly interested to see whether anything could be done about improving this characteristic of pod shatter and making these pods more resistant to shattering. So we identified a target gene that we thought might have this desired effect, and we went ahead and carried out gene editing to introduce a specific mutation in the target gene. So I've got some detail in the slides, which I'm not going to go through in great, but it's just there if anyone's interested. But basically, we targeted this gene, GA3. There were 2 copies of this gene in the brassica genome. There's an A copy and a B copy, and we just targeted the A copy. And that's important because I want to talk to you just a little bit about off-target effects in a moment. But so we targeted the A copy. So we designed these specific guides to target the region that we wanted to. We assembled all the components into a construct, and we went through the standard transformation process, introduced these into brassica. And this is what we saw. So this is the wild-type plant, and this is the edited plant. Now, we were expecting it to be dwarf, and it is, but I want you just to look at this section through the seed pod. So this is the section through the 2 valves of the pod where they come together. There's a region called the valve margin. And you can see that valve margin here in this wild-type section. And you can see there's a small groove, and this is where these 2 valves are going to come apart. And if you look at the edited plant, you'll see a completely different structure here. And this valve margin has been changed. So obviously these are going to stick together much better, and it's going to be more resistant to pod shatter. So we did get the outcome that we were hoping for. And you can see that was due, this is just the DNA sequence of the target region. You can see the small change that's happened in 2 alleles of the 2 mutant alleles here. There's a single base change and a single base deletion. It's a very small change, but it's giving us this change in phenotype. So it's also important to say that in the next generation, we're able to isolate. So although we've introduced all the components in a genetic modification process, we're able to segregate away, with just normal genetic segregation, all of those extra genes in the next generation and end up with a plant which is homozygous for the mutation that we want, but it doesn't contain any of the components, any of the transgenes. They've completely gone, and we can see that here. So we have a edited, transgene-free plant. I did mention that actually in this example there were 2 copies of this gene, an A and a B copy, and one question that often comes up is there any off-target activity? So if you target one gene, are you actually going to inadvertently target another one? And so the answer is it depends on the design of your guides, but we did see some off-target activity. And you can see it actually, so in this example here, where the B copy just had 2 base changes compared to the target sequence in the A copy. And we did see a little bit of off-target activity. But actually, in crops, this has to be viewed not only as it's not really a problem, it's a real opportunity for us, because crop genomes can be very complicated. They can have multiple copies of genes in gene families, and often you want to target multiple genes in a gene family. And this technology does give us that opportunity to do that with a single guide. Okay, so another example I just want to mention. So one project that we're involved in, this is a Gates Foundation funded project. It's called ENSA. It stands for Engineering Nitrogen Symbiosis for Africa. So this is a very ambitious project, and the aim is to try to engineer the cereals so that they can carry out some nitrogen fixation. Now, cereals don't normally do this. Crops like legumes are very efficient at doing this because of the association with the bacteria that allows them to form these root nodules and to fix nitrogen. So this is a very ambitious and quite a long-term project. But I just wanted to highlight to you that actually the gene editing is making a real difference in a project like this, because it enables us to look at some of the pathways that are really important, that we understand them and that we understand which genes we need to modify and engineer. And it enables us to knock out the genes in a pathway one at a time. So, for example, in this symbiosis signaling pathway, we've actually currently knocked out 14 genes. We've got some more going through. And this gives, and we've done this in barley, and this gives us a really valuable research tool to begin to tackle this really big problem of can we possibly get some nitrogen fixation into cereals. And this could make, even if it's at a very low level, it could make a huge impact, especially in Africa. So just to show that this is, as a research tool, this is incredibly important. I'm not actually going to go through the strategy of how we did this, but just to say, I'm going to just skip this if anyone's interested. You can look at the details, but I just want to show you how efficient it is. So for these 14 target genes, what we're looking at here is just the percentage of the primary transgenic, the primary plants that we produced, what percentage of them actually had this genome editing active in them. And you can see that it can be quite high. Here we're over 80%. The average is between 30-40%. So it's a fairly efficient process, and we were actually successful in editing all of these target genes. We also were able, because our standard technique is to use 2 guides in each construct, and we can then ask the question of were they both active or was one just active. And we can look at that here. And sometimes in a construct just one works, sometimes they both work. And actually out of 25 different DNA constructs for gene editing, only one of them failed completely. So just to give you a feeling of how efficient this is in the crops. Now I have one more example. Sorry, this looks a bit complicated, but this is an example in wheat. And I just put it in because this was a real challenge, but just to show that actually it was still possible to get the outcome that we wanted. Graham Moore's group at the John Innes Centre have been working many years on this particular gene locus called PH1. Now in wheat is a complicated beast. It has what we call a hexaploid genome. It basically has 3 ancestral genomes combined in the bread wheat that we currently enjoy. It's A, B, and D. And so it has pairs of chromosomes which are homologs, but also it has chromosomes which are homologs, which are the similar ones but on the different genomes. And this locus, so wheat, if these chromosomes don't pair properly, then wheat is going to be in a bit of a muddle during meiosis, in cell division. It's not going to work properly. So this locus is responsible for controlling the pairing. And Graham really wanted to target one particular gene in this locus, but only on one chromosome, and that was on chromosome 5b. So there's also copies on the group 3 chromosomes. He didn't want to touch those. So we had a look at this to see whether it was possible. And actually, yes, it was possible, but we were very limited. We could only find 3 possible guides that we could use, one which was quite good and 2 that were okay, this is just enough variation. So we went ahead and we tried. We used the best guide paired with one of the others in 2 constructs. We put them into wheat. And we actually screened just over 80 plants, and we found 4 that had active editing. So that's only 5%, which is much lower than the other examples. But we were expecting this to be difficult. But we found what we wanted, and particularly we found one plant which had a perfect deletion of the region between these 2 guide sequences, so 115 base pair deletion. These were then taken through to the next generation, and we were able to find homozygous plants with this specific deletion in this ZIP4 gene, which was exactly what we were looking for. So this is important because wheat breeding, there's a real challenge because of this control of chromosome pairing. So for the wheat community, this is potentially quite important. And you can actually see the impact of this mutation in these chromosome spreads here, looking at these meiotic configurations. So this is the CRISPR mutant, and this is the control. And this is something similar done with another technology called tilling, which is a random mutagenesis technique. And you can see that the tilling mutant and the CRISPR mutant are looking very similar. So this was just a nice example of we could actually tackle quite a difficult task. So there are many nice examples now in the literature of using gene editing in crops. So one in particular, so things that I think where there are real opportunities. The first is disease resistance. This is a very nice example from the lab of Taisha Gao in China, who was able to knock out 3 copies of this gene MLO to confer resistance to mildew, which is an important disease. This was actually done initially using talons, but again, a gene editing approach. There was a similar example, another disease resistance in rice, tackling a very destructive rice disease called rice blast. again, using CRISPR and showing significant resistance to rice blast, which is quite exciting. And this one you've already seen, the tomato example. Again, faster flowering, more even yield is another possibility. So in my lab, we have something we're working on at the moment is engineering drought tolerance using CRISPR in cereals. I don't have results to share with you yet, but I do believe that there are real opportunities to use CRISPR for engineering drought tolerance. It's something we're looking at. So what's in the pipeline? Well, this is just a table showing, it's from the USDA, just showing CRISPR-edited plants that are in the pipeline, and the USDA has decided that they do not need to regulate these because they've just got small mutations. These are CRISPR mutants. So in the United States, these are all going, these are in the pipeline at the moment, things like this non-browning mushroom and a waxy corn here. I'm not going to say anything much at all because I know Joyce is going to talk hopefully about regulation, but just to say that obviously in the EU this is currently being considered by the European Court of Justice. Just earlier this year that we got the first opinion from the Advocate General, and just a little quotation from it here saying, so organisms obtained by mutagenesis are in principle exempted from the obligations of the Genetic Modified Organisms Directive, which I take as a very positive statement. It's not, there's a lot of discussion still to be had around this, but we still await the final decision of the European Court of Justice on this. And of course the decisions made on how these technologies are regulated are vitally important for taking this technology forward. Now I just want to mention in my last couple of minutes, we've talked a lot about using CRISPR to make targeted mutations, small insertions or deletions, but there's a whole raft of other things that can be done as well. And again, most of these have been done first in animal cells, but they're now moving into plants, and and they're working very well. We're trying a range of some of these things currently in the lab. So, as well as the standard Cas9, you can get different versions of Cas9. So one that will make a single strand break, and one that is actually a dCas9, which you think of as a dead Cas9, that doesn't break the DNA at all, but it's able to be fused to other proteins and take them into a targeted location. And this is really very valuable because then you can look at new ways of upregulating or downregulating genes. You can even look at doing what we call base editing, making a specific base switch. So with this case, fusion to a cytidine deaminase, you can do a CT switch. So if you want to do something even more precise and just change a single base, it's possible. There's also opportunities for changing the methylation status. So there's many more opportunities. I think we're really at the beginning in the plant world. The ability to make precise insertions is also incredibly important. And this is something, this is something we've just started to look at in crops. And I think my message is that it is working, and it's actually working better than I expected it would. So in plants, once you've got the double strand break, what normally happens is this error-prone, this non-homologous end joining repair process kicks in, and that's what normally creates the small mutation, the knockout. A far rarer process is this repair by homologous recombination. And that's what you need to allow precise gene insertions. So we've actually shown, using this sort of scheme here, we've actually just inserted in a precise location a fluorescent marker gene in barley and just shown that we can do this, and we've gone through to the next generation and found some very strong bands showing precise, and these have been sequenced, precise homologous recombination events that we were expecting in barley in the next generation. And now we're going through another generation to look at inheritance. So this is just to sort of show that actually this, and this type of technology is very important for so for example, if we want to engineer for disease resistance, we don't want a single disease resistance gene. We need a number of them, and we need to stack them at a particular location. Very important in crop breeding. And this sort of technology will allow us to do that. So now, just right at the end, come back. You remember my plate of food here. This was 2015. This was last year. This was actually the first CRISPR conference dinner. Now, you may notice that it looks slightly more sophisticated than the first meal, and I actually like this. I like this comparison because to me it really shows how this technology is developing. This was also served in Sweden, and it was a conference dinner for an editing conference. So I'll leave you with that, and I just want to particularly just acknowledge the people in my group. Tom Lawrenson, who's in the photograph. My pointer doesn't seem to be working now, but in the blue T-shirt has done a lot of the editing that I've described today, and the other members of the group made a huge contribution. Thank you.
Well, again, many thanks, Wendy, for a great overview, and maybe not surprising, many of the opportunities, many of the challenges are exactly the same as we face in the labs on a daily basis trying to generate models of disease. I have, though, learnt that what we need to do next time round is arrange a CRISPR meeting at the AstraZeneca site in Gothenburg, where we can then share the food.
Yes.
We have time for a couple of questions, but maybe I'll just kick off. Again, you did allude to off-target effects and actually describe the benefits of off-target effects. We worry very much about off-target effects in terms of therapeutic editing. How much work do you do to demonstrate there are no off-target edits in the crop species that you're working with?
So yes, that's a good question. So at the moment, we've really only looked for the off-targets in the most obvious places where we might expect them. If we want to address the wider question of are there off-target effects throughout anywhere in the genome that we simply couldn't predict, then our only option is to sequence entire genomes, which of course we are now able to do. So I think that is something that we might look at doing, just to give us the confidence that that we can answer that question and say, actually, yes, we have looked extensively and there is not anything else. Yeah.
Mark?
I think as we push the CRISPR area forward, one of the most important things is public engagement. And probably in this area, the public are most sensitised to GM anyway. And I just wondered what initiatives you were involved in, which was reaching out and trying to get over the more controlled approach to change than what was there before.
Yeah, no, it's a very good point. And I think our approach is just to try to be completely open. If anyone wants to know what we're doing, we're very happy to show them and to try to explain the technology. We've tried to look at ways of explaining the technology in a very accessible way. For example, we were involved in a local science festival where we had hundreds of people coming through, a lot of school children, and we just tried to do it, actually have it as a simple demonstrating CRISPR as a treasure hunt to find the target sequence. And then, and that went down quite well. I'm not sure how, but we've, just taking the opportunity to just talk to as many people as we possibly can. We're also looking at some new videos that we can possibly make available to try and just to make as much of the information available as we can. It's a challenge though.
Hi there. On a similar theme, Jonathan Leake, I'm the science editor at Sunday Times. And I was around when GM came along and also when the backlash came along, which was in some senses driven by a perception that GM had a lot to offer the industry but not so much to offer the consumer. And what I've heard in the last two talks is some fascinating and brilliant breakthroughs, and I can see why all of you are excited about it. But what would you say to consumers in terms of the benefit they're going to reap from this? What are they actually going to see that wards off, if you like, that sense of consuming something that's a bit alien?
Okay. So I think there are examples certainly coming through that are more what you would call a consumer benefit, a direct consumer benefit, where you're looking at maybe nutritional benefits. You may be looking at using CRISPR technology to remove a toxin or some sort of anti-nutritional, and this is possible, and it might well be something that there's a lot of demand for. An example in wheat, so there was a publication to try to produce a gluten-free wheat, and that's very close. So all the people who have a gluten intolerance or an allergy, this could be something that they would certainly want. So I think those sort of examples might help, but I think it's down to us to, just to try to make the case for some of the other modifications that we're making, which are really addressing quite critical issues of disease resistance, although that's not so directly applicable to the consumer. That we're talking about the future of our food supply, so it's still pretty important. And it's just getting those messages across, I think, in the best way that we can.
I think that's a great question. Again, maybe Mark, Sarah and Robin, in the ethical debate towards the end of the day, another great question. How do we describe the benefits of these technologies to the consumer? With that, Wendy, we need to move on. Many thanks again for a great overview of the potential impact here of CRISPR in crop science.